Assalamualaikum sir, this is Mahfuzur Rahman Jami and my ID number is 1920809.
Assalamu alaikum. I am Rizwan Hussain, 19208019.
Assalamu alaikum. I'm Science Economy and my ID is 19208. For giving us the opportunity to present our presentation. So today our presentation is about UVC disinfect SARS-CoV-2 by inaction of viral genome damage. So first of all, we have to know what is UV. So ultraviolet is a form of electromagnetic radiation with wavelength shorter than that of visible light and longer than X-ray. UV radiation is present in sunlight and constitute about 10% of the total electromagnetic radiation output from the sun. So this is a very short wavelength. It has a very short wavelength. So short wavelength ultraviolet light damage DNA and sanitary surface from surface with which it come into contact. For a human, suntan and sunburn are familiar effects of exposure of the sun of UV light, along with an increased risk of skin cancer. On the other hand, ultraviolet visible spectroscopy is widely used technique in many areas of science, ranging from bacteria culturing, drug identification, and nuclear acid purity checks and quantitation to quality control in the beverage industry and chemical research.
Virus and cells. Infection medium minimum essential medium equal MEM thermoofficial scientific with 2% FBS 1% PSG was used to infect Vero E6 with the SARS-CoV-2 virus. In Vero E6 TMRSS2 cell SARS-CoV-2 was propagated for 3 days at a multiplicity of infection MOI equal to 10-5. Utilizing TCID 50 tissue culture invective dose assays virus were treated. In order to perform the TCID 50 assay, Veros E6 TMP SS2 cells were placed in a 96 well plate with 104 cells per well. 100 liter of 10-fold serially diluted virus containing infection medium was used for infection, and each dilution has 8 replicas. The cells were then incubated at 37 degrees Celsius for 3 days. The virus-induced cell cytopathic effect was used to determine viral infection in each well after incubation. The read mutant formula was used to determine virus titles 4-9.
This materials and method. So virus culture and UVC irradiation. In this process, a culture of the virus grown into a stable host cell. Once the virus reached a sufficient concentration, it can be objected on UCV, ultraviolet irradiation. UCV irradiation is a method used in inactive virus by damaging their genetical materials. Inactively assays. These assays are used to determine the ability of a virus to infect host cell and replicate within them. Virus techniques can be improved, such as where the virus is serially duplicated and added to a morphology of host cells. Immunicating analysis. In immunicating analysis, also known as Western bottling, is the technique to use, detect, and analyze, sacrifice protection, and in samples. In the context of virus researches, immunity protection can be used to identify and quantify the virus potential and the other things based on their size, their gel, electrons, and etc. Transmission transmission electron microscopy. This is a high-resolution imaging technique that allows the visualization of virus protecting their agriculture details with them. Research can examine the morphology, size, and internal structure of a virus. In my five number point, we have the development of a lengthy RT qPCR, which is reverse transcription contributive priority chain reception, is a technique that detected the quantity of specific RNA or DNA species in a sample. In the text of geomet damage assessment, we are all from this slide. In my next slide,
Virus culture and UV irradiation. UVC irradiation is a method used for disinfection and sanitization. UVC refers to a specific range of ultraviolet light with a wavelength of 200 to 280 nanometer. This range of ultraviolet light has germicidal properties and can effectively inactive or destroy microorganisms, including virus, bacteria, and fungal. UVC radiation damage the DNA or RNA of the microorganism, disruption their ability to replicate and causing their death and inactivation. So there are some steps to do this method. These steps are given here. Sample collection, inoculation, incubation, then viral replication, harvesting, purification, and storage.
next sample collection, which is sputum collection for individuals with respective symptomism. A deep cough may produce sputum, which can be collected for testing. Refer to much and other materials that is caught up from the lower respiratory test.
Isolated virus is introduced into a suitable host cell culture. Viruses are obligated intracellular parasites, meaning they require a host cell to replicate and propagate. Host cell can include animal cells, bacterial cells, or even specific cells line delivered from human or animal tissues. Incubation. The virus-infected host cells are placed in a controlled environment, such as an incubator with optimal conditions for virus growth. These typically include maintaining appropriate temperature, pH, and nutrient supply to support virus replica.
Vital replication. Inside the host cell, the virus uses the cellular machinery to replicate its genetic material and synthesize vital protein. This leads to the production of new virus particles which can infect neighbor cells. When microorganisms or virus are exposed to UV light, their nucleic acids are damaged in a way that they are rendered incapable to reproduction and infection. Bacteria absorb light. Bacteria or virus absorb light for growth process. Therefore, high energy UV light is absorbed as well and entered cellular RNA and DNA. This absorption of UV energy form new bond between adjacent nucleotide, creating double bond of dimers. The longer the exposure of UV light, the more thymine dimer are formed in a DNA, and the greater the risk of a incorrect repair of a missed dimer. If cellular processes are disrupted because of an incorrect repair of remaining damage, the cell cannot reproduce and infect anymore.
The harvesting system of SARS-CoV-2. SARS-CoV-2, the first sampling collection. First is sampling collection. We need to collect the sample first, as mentioned earlier in my last slide. Then the sample processing will be held. After that, I think the that collected the sample are transferred to a laboratory where they undergo various processing steps that typically involves transferring the collected sample into a suitable transport medium, which helps maintain the viability and stability of the virus during the transformation. The next step of the sampling is sample inactivation. To ensure the safety of laboratory and other minimizing the risks of the virus spreading, collecting the samples are often subjected to infection protocols. The infection treaters, the treating of these samples can be too harmful. Next step is viral RNA execution. The inactivated samples are the process to execute the viral RNA. The step involves breaking open the viral particles and involving the RNA using the version of extraction method. The next method is reserve transformation and amplification. Once the viral RNA is extracted, it undergo reserve transformation where a RNA is converted into a complementary DNA. Then the last step is detection and analysis. The amorphil of DNA fragmentations are analyzed using the various methods such as the real-time CPR and other things that used in SARS-CoV-2 generally. All of this. Additionally, analysis the inclusive genetics and squishing and identify sacrifice variant of multiply the virus.
The harvest virus containing fluid is then subjected to purification step to remove cellular debris, host cell components, and other contaminants. Purification methods can include centrifugation, filtering, and ultracentrifugations or chromatography techniques.
Storage. The purification virus is stored under appropriate conditions, often at low temperature, until further use or analysis.
Results. Time-dependent inactivation of SARS-CoV-2 by UVC radiations. Preservations of viral protein integrity after UVC treatment. Morphological examination of UVC-treated virus particles. Evaluations of genome damage using the newly developed lengthy RT-qPCR test.
Discuss about the discussion of SARS-CoV-2. There are so many discussion in my slides. First, I am going to share the first line, that is Mechanius underlying UCV inactivation of SARS-CoV-2. And my second line is, the second point was implementation for virus transformation control and the inactivation parts. The use of UVC irradiation has important element of in virus transformation and control the interference prevents UVC can be applied to disinfect virus surface and objects that cover SARS-CoV-2, including personal and prevention medical technique and high-tech surface healthing, and UVC diffusion can help reduce the risk of virus to spread out. And the third line was the comparison of UVC efficiency with the other disinfective methods. UVC disinfection, there are so many UVC disinfective methods, but here we are using the ultraviolet ray, which is very good for the SARS-CoV-2, and it's used for the coronavirus throughout the last few years. In my last topic, I am going to discuss about the limitation and future direction. While UVC irradiation shows promises as the diffusion method for SARS-CoV-2, it has come to limitation and areas for future intervention. One limitation is need for direct impulse of UVC light to the virus surface and achieve the effective irradiation. That's all for my discussion and my presentation, and that's it for today. Assalamu alaikum.